Hey everyone, welcome back to Strange New Worlds, a science and Star Trek podcast. I'm your host, Mike Wong. As many of you know, I am a professional astrobiologist, and I have an exciting new scientific paper coming out. This time we're looking for signs of life in ancient rocks. It's honestly the kind of study I couldn't have even imagined being a part of, much less lead author of, a few years ago. You see, being a Star Trek fan for my entire life, I've always been much more interested in what was above my head than what was below my feet. But this particular science project showed me that looking down and back in time can be an important step toward the future of how we study worlds beyond. Let me tell you a little bit more about what we've done. We are essentially teaching computers how to identify molecular signs of life across Earth history, from the Archean eon some 3 billion years ago to today. If I had to distill our findings into 3 key takeaways, they'd be, number 1, we have now doubled the age with which we can use suites of organic molecules to identify signs of life in ancient rocks. Now, scientists have developed many ways to infer life in ancient samples. You can look at the textures of rocks, the minerals in the rocks, the isotopes of carbon. But unambiguous records of life using complex molecules extends only back to about 1.6 billion years ago. But now, we take that age all the way back to 3.3 billion years ago. Second, we demonstrate that our biosignature technique, which utilizes machine learning, is not only capable of discriminating life versus non-life, those 2 broad categories, but also different kinds of life through deep time. In particular, we place an emphasis in this paper on identifying signs of photosynthetic life, which we find evidence for in rocks as old as 2.5 billion years. And third, we show the power of information as a vital parameter in the natural world. You see, some of these oldest rocks on Earth have been physically degraded and their molecules chemically altered over billions of years. So not a single original biomolecule from any living cell is left intact. Yet, we can still detect the echoes of biology by looking at the information that life has left for us in the distribution of organic molecules in any given sample. This holistic approach is how we are able to look for clues in time capsules billions of years old. Now, to tell us more about the details of this work, I'm beaming aboard my co-first author, Dr. Anirudh Prabhu. Like me, Anirudh is a research scientist at Carnegie Science's Earth and Planets Laboratory in Washington, D.C. He specializes in developing and applying artificial intelligence to making discoveries about the natural world. Anirudh and I have partnered over the past several years on numerous projects, and this work is some of the most exciting stuff we've ever done. It's attracted a fair deal of media attention, and so to prepare for the interviews that we had with journalists around the world, we thought we'd kill 2 birds with 1 stone and record a podcast to share with all of you right here on Strange New Worlds. Ready? Engage! Welcome back to Strange New Worlds. This is an emergency podcast because we have a very exciting scientific paper coming out on November 17th, 2025. So when the podcast drops, that should be the day that the paper is actually out, so you should be able to go online. I'll put a link in the show notes to our paper. It's being published in the Proceedings of the National Academies of Sciences, or PNAS, and we're recording this podcast a few days before the paper comes out because we got invited to do a radio interview, and my co-first author on the paper, Dr. Anirudh Prabhu, a research scientist at Carnegie Science's Earth and Planets Laboratory, is going to take that radio interview, and this Emergency Strange New Worlds podcast is just to practice that. Welcome aboard, Anirudh.
Thank you. Thank you for having me. And I love the impromptu nature of this. This will be really fun.
Yeah, it's going to be great. And the beautiful thing about podcasting is that you can stop and start as many times as you need. So this truly is a practice, but the
Good.
audience will only hear the best part.
Glad. I'm glad the editor likes me.
Okay, so the title of our big splashy paper that's coming out in PNAS is called Organic Geochemical Evidence for Life in Archean Rocks Identified by Pyrolysis, GCMS, and Supervised Machine Learning. So basically, our study targets looking for signs of life or biosignatures in ancient rocks on Earth, rocks that are as old as 3.3 billion years old. But hold on a second, Anirudh, aren't all rocks on Earth billions of years old?
Well, no, because Earth itself is very old, right? It's 4.5 billion years old, but it's a pretty active planet. There's a lot of tectonics going on, volcanic eruptions happen. And so every time, say, a volcanic eruption happens, it puts a bunch of more rocks out on the surface of our planet. And so rocks have pretty different ages depending on when they came on the planet. So not all of them are billions of years old. Some of them are pretty young comparatively.
We gathered our data for this study by running 406 modern and ancient samples through a chemical technique called pyrolysis gas chromatography mass spectrometry, or for short PyGCMS. For non-specialists in the audience, which are probably all of the audience, how does PyGCMS work and why was it key to this study?
Well, I'm going to steal the way you describe it. It's an oven that bakes your cake and tastes it for you too.
Yeah.
So a pyrolysis GCMS has three broad components. I'm not going to go into too much detail, but the first is the pyrolysis part where you heat up the sample, slowly weakening its bonds. The gas chromatography part separates the molecules out, and then the mass spec actually is able to produce spectra that help you identify what these molecules are in these samples.
So in our study, we essentially want to distinguish the chemical fingerprints of life from the fingerprints of things found in non-biological materials, such as meteorites. So how does AI power this kind of discovery? What does an AI model look for to detect a biosignature?
Yeah, that's a really good question. Think about the data sets that are being produced from every sample as a data landscape, right? So you have a vast data landscape. There are mountains, there are hills, there are peaks, there are valleys, there are so on. The AI model is able to identify features in this data landscape that repeatedly occur and use patterns in those features to identify whether a sample is biotic or abiotic. We can't quite tell from these detailed complex features by eye whether something is alive or not, but by training an AI model that's repeatedly seeing a set of peaks occur in a particular area in this vast data landscape, that, okay, that's biotic, or that's never been alive, that's a meteorite or something like that. That's how an AI model is able to look at repeating patterns.
So connect the landscape idea with an actual sample. So let's take a sample that's clearly alive, like a leaf. Mm-hmm. We put it through the pyrolysis GCMS. It produces some kind of data landscape. We take something that's never been alive, like a meteorite. It produces some other data landscape with these peaks and valleys that you described. What do those peaks actually represent in those samples? Why are they different between the leaf and the meteorite?
Right. So if you remember my description of the PyGCMS, you heat up the samples and different things burn at different times and different bonds weaken at different times. So say in a leaf, a lot of things are burning in a certain area. This is going to be true for things that are chemically similar. So all things that are new leaves are going to be behaving in a similar manner. In fact, the example that you gave, the leaves versus the meteorites, are some of the easiest things to distinguish because they're so different from each other. Unlike if I try to compare a leaf to a root or something like that, that's going to look much more similar. So harder to distinguish in a manner, even for us, let alone for AI.
Well, let's talk about distinguishing things within the living category itself. Mm-hmm. So can we find out about different kinds of life? How does that work?
So we created 9 broad categories of samples for our 406 samples. So we are looking at directly photosynthetic, indirectly photosynthetic for modern life.
Break that down for the audience. What does that actually mean?
So what that means is, like, if I pick up an actual leaf, things that has chloroplast in it, where it can actually perform photosynthesis directly, then we're labeling it as directly photosynthetic. If you're picking up non-photosynthetic parts of plant material, roots, stems, bark, things that are far away from what we know as bright and green and, can take in carbon dioxide, put out oxygen, that kind of thing. We're calling it modern plant, but not photosynthetic. We have similarly bunch of other categories within life, right? We have things that are not plant material, that are animal material. We have things that are ancient life. So they used to be ancient plants. They used to be animals a long time ago, but they're all now fossilized in some way or the other. They've gone through a lot of processes. All of these things are life as we define it broadly, but there's so many subtypes there, right? And so that's what we're trying to do, is to make these groups to see if our model can actually tell the differences between those. And so far, for the most part, it can, which is really encouraging to see.
One thing that was very astounding in our paper was the report of photosynthetic life in 2.5 billion year old rocks. Mm-hmm. So tell us about that discovery. How did we determine that there was actually photosynthetic life, not just life, but photosynthetic life? And what does that mean more broadly for understanding the history of the Earth?
Yeah, so this is, again, one of the most baffling things we found in this paper. We had a bunch of models that we tested out broadly. One of our 4 major models was a model that could distinguish photosynthetic versus non-photosynthetic samples. And just like I described, the pattern recognition happens in this data landscape. And as part of this, we trained a lot of current life and ancient life as photosynthetic. We labeled them as photosynthetic and trained our model to identify previously unknown samples. So these are samples that are hotly debated in the field. They have been for a few decades now, if not longer. And there's a group of people saying that used to be photosynthetic. There are others that are like, that's not what we think, So we had about 65 or so, I'd like to think, I've got to remember the number, truly unknown samples that we ran our final model on. And in that, we found some really cool results. The oldest of which is this formation, which is 2.5 billion years. And we were able to predict that it is photosynthetic. Our model had a 58% probability that that particular sample, which was from South Africa, I think, was photosynthetic whenever it was alive. The second part of your question was what was your second part of your question?
It was more about Earth history. So let me just recap what the major findings in the paper were. So we say we detected signs of life in 3.3 billion year old rocks, and we detected signs of photosynthetic life in 2.5 billion year old rocks. These findings extend our molecular evidence for photosynthesis by roughly 800 million years and roughly doubles the time interval for molecular evidence for life itself on planet Earth. So given these major findings, how does that change the way that we understand the coevolution of life and Earth?
It changes it significantly, at least I'd like to hope so. We can now identify the processes that existed with a bit more confidence than before through these molecular biosignatures. So as you said, we've sort of jumped quite a lot in how far back we can go to identify molecular biosignatures. So the oldest molecular biosignature, I believe, was 1.5 to 2 billion years old, right? That's what they were able to find. And beyond that, you had to look at structure, you had to look at isotopes, you had to look at some other features. You couldn't tell whether it was alive or not back then, purely based on the molecular signatures. The AI model is able to take these sort of degraded, diminished signals and still make sense of them because there's still some information stored in these samples. So imagine you have the same data landscape with a lot of peaks, but it's really an old degraded samples. Everything is a little muted, everything is a little diminished. And just because the spectra are diminished and muted doesn't mean that the relationships between the peaks and the landscape structure itself doesn't change. And so it's still pattern recognition. The model is still able to pick up what truly the sample is. And so what I'm hoping our contribution does is for people to study some of these older samples through the lens of an AI model to identify what are the most interesting samples, whether it was alive or not, what are the processes of formation. And suddenly a whole new set of data is at our fingertips and analyzable that we just had set aside before.
Now, our approach marries traditional chemical analysis with state-of-the-art artificial intelligence. Everybody by now knows that AI can make mistakes. Facial recognition can be biased. ChatGPT hallucinates famously. So how confident are we that our AI model identifies truly biological activity when it says it does?
Again, a great question. I think as people creating the model, we're the most skeptical of our own results more than pretty much anyone in the world. So every one of our predictions has a probability associated with it. It has a confidence value to the predictions that are made. We used a random forest model for this particular iteration of our paper, and it's also one of the more interpretable models where we can get in and see what are the features contributing to each prediction. So I would say, based on what we've seen, we're pretty confident, but I would like every one of our predictions, every one of our results to be experimentally tested, to be independently verified. I would not say with full confidence that anything is alive or was alive without it being independently verified like 15 different times.
You're speaking to the mantra in biosignature research of multiple lines of evidence. When you want to make a claim of life, especially ancient life, or maybe even alien life one day, we want multiple independent lines of evidence to all point to the same thing.
Absolutely. That's the mantra for every single scientific finding that we make. So if you ask me how confident I am that a sample is alive, I can't quite put a number on it. How confident is the model? I can tell you for every single prediction how confident it is, and that number changes.
Yeah. And what we've offered the community essentially is another arrow in our quiver to try to understand the most enigmatic samples and where they came from.
Exactly. And, paleobiology is rife with data, and this is just another toolkit in their belt. Exactly how I see it, yeah.
Now, something that raised my own personal credence in our own model was the surprises that it gave us.
Yes.
Anirudh, would you tell us about some of the samples that the model classified differently from what we expected?
Yeah, so there were a couple of surprises at the very least, but let me give a little bit more context for the listeners here. So there are certain samples that either are wrongly predicted or just don't behave the way we expect them to behave based on the data and based on the knowledge we have of the sample. And so we check these pretty routinely and many times over. So there are certain things that just didn't run right? So like a particular rice sample didn't run and it produced a weird spectra, so we reran that sample, something like that. But there are these repeated samples that we kept coming across that were wrongly predicted in our initial runs. My favorite example of the lot is the wasp nest example. So we had a wasp nest that we ran through our PyGCMS machine. And we didn't exactly know what a wasp nest is made of, but we initially labeled it as non-photosynthetic, right? We're like, well, it's a wasp's nest and it's non-photosynthetic. And we labeled it and we proceeded with our models and we ran some tests and our model kept getting it wrong. So we were wondering whether this was just a dud sample, whether there's something wrong with this run, whether Mike, who was running some of these samples, did something wrong. But when a model is confidently predicting it, you also have to account for that there's something actually in there that we didn't know about that is leading to this prediction. So we eventually came to that step of checking why a wasp nest is always coming up as photosynthetic. We just didn't know much about how wasps make that nest at that time. So we dug into the literature. We tried to find out more about the wasp nest that we had and that we ran. And they were like, okay, so wasps apparently build their nests by chewing up parts of trees and sort of using that to construct their nest. So there's absolutely tree material there. There's absolutely photosynthetic material like leaf material there and so on. You can't quite tell when the final product is done. So we didn't know. But the model knew. It was able to find that the wasp's nest and its molecular compositions were much closer to all the photosynthetic material than it was to anything non-photosynthetic.
That's so cool.
Yeah, I mean, we were blown away. There were a couple of other examples. I think you mentioned an example yourself that was interesting, right?
Yeah.
The sea squirt. Let me throw this back to you because you had another interesting example of a surprise that we saw.
Yeah, so look, a sea squirt is a type of marine animal, and so clearly not photosynthetic, right? But in our model, it kept on returning a class probability of photosynthetic of 71%, which is way above our threshold of roughly 60% to, confidently say that something is photosynthetic. And we were like, what? Why is the sea squirt shell coming up as photosynthetic? It's like literally the shell of a marine organism. And then we learned that sea squirt shells are often coated with photosynthetic algae. And so this was, again, a time when the model was way smarter than us. It knew that the sea squirt was coming up as photosynthetic. And then we had to actually go and look at the literature and be like, oh, it actually kind of is photosynthetic because of the algae that grows on it.
I love when these kind of things happen when the model challenges us in a way or challenges our knowledge. Not only do we get to learn a lot more, but it gets us to be more accurate in the next iteration of the modeling.
Absolutely, absolutely. Now, the things that we discovered about sea squirts and about wasps' nest, that was just things that you and I personally didn't know about, but humanity has figured that out. We were able to Wikipedia it very easily. But hopefully one day we're going to launch this type of technique to discover something about organic molecules on another world that nobody's ever known about before. So you and I are both really interested in astrobiology. Eventually, we want to develop this method so that we can take it off Earth and make discoveries about everything else out there. Anirudh, how do you envision applying this method to the search for life elsewhere in the universe, be it Mars or Jupiter's moon Europa, or somewhere else?
Like a PyGCMS is already out there on space missions, and it's one of the most flight-ready instruments that we have. And our method is extendable to pretty much any PyGCMS, but really anything that produces a spectra. But that's a different project that we have going to test all of this out. So what I envision is, if a sample is picked up and analyzed through a pyrolysis GCMS on another world, we can add that spectra to our data and have it tested out to see what that particular sample is closest to in our training database. And if it's closest to life as we know it on Earth, wonderful. We just have made our first prediction for life on Earth, in other worlds. But if it isn't, it still tells us more than we currently know about some of those worlds by telling us, relative to everything that we know here, how does this new sample rate?
So science fiction, and especially Star Trek, is rife with examples where life elsewhere, alien life, does not use the same chemistry as life on Earth. So do you think that our approach can detect life that uses different chemistry than life on Earth does?
The short answer is yes, but of course there's an asterisk on that to say why I mean that it can. Our model can figure out how a new sample relates to everything else in our training data, right? So while it can figure out that this particular sample that we found on another world is closest to life on Earth, or is closest to non-life on Earth, is closer to a meteorite and nothing else, it can also figure out that actually this new sample you picked up is unlike anything we've ever seen before. It's unlike any life or non-life that is in your currently 406 samples, but we're expanding rapidly towards 1,000, right? That's unlike anything anyone has seen. So our model can perform what's called anomaly detection. It can detect an anomalous sample. And to me, the most exciting part of astrobiology is not even getting to the part where like, oh, this is life or not. It's identifying an anomaly and saying, oh my God, this is unlike anything we've seen before. We got to study this. We got to get more information about this one way or the other. And our model has the potential to do that. So that's probably the most exciting part for me.
I can't wait for that day to happen.
Yeah.
What are the next steps for us in terms of getting where we want to go eventually?
Right. There's a few next steps, short term, based on the hurdles that we faced in the current project. Obviously, we want to add more samples to get a bigger data set, but we want to add these samples strategically and specifically because we have what's called a class imbalance problem in machine learning. So remember I told you we had 9 categories, right? In those 9 categories, we had a couple of categories that we don't have enough samples on. We don't have enough, I would say, diverse set of fungi. We don't have enough fossilized animal samples, I believe it was. And we want to add more samples to this so our model gets better at identifying the landscapes that are repeating for those kinds of samples. So that's one thing. The second thing is obviously to interpret the results of our predictions a bit better. As I said, our random forest model is sort of inquirable and interpretable and explainable. So we want to dig in to see what actually makes photosynthetic samples fundamentally different from non-photosynthetic samples, or ancient life different from ancient plant life, different from ancient animal life, things like that. And by digging in, we can at least try to understand the reasoning involved. And I'm drawing quotes for people who can't see me. Reasoning involved in making a particular prediction or a particular choice. And this is quite commonly done in AI-based research because you want to understand why a model made a prediction just as much as you want the model to make an accurate prediction.
Excellent. Philosophically speaking, what does it mean that the traces of life can still be quote unquote heard 3 billion years later?
I'd like to think that, so this goes back to the example I gave you about the diminished data landscapes that are created, right? So everything on Earth, all the rocks on Earth, everything that we have, especially these ancient samples, hold a ton of information in them. Just because something is very old and that information gets degraded or diminished doesn't mean that there's still not information there for you to extract and explore and understand. And so when we're saying we're hearing the whispers of the traces of life from 3 billion years ago, we're really saying there's still information in these very ancient samples that we can use to learn about what the Earth was like back then and to add to our knowledge on what happened on Earth around then and how things evolved from then.
Does our research change the way we think about the persistence of life's signatures in the universe?
I'd like to think so in a short answer. The longer answer is going back to, the information stored in the rocks. Part of what we do, part of what I've been doing for close to a decade now is studying the evolution of minerals on Earth. And I always call them like USB drives, right? They store all the information of everything happening around them. That includes the environment, that includes life around it, and so on. So life has been persistent on Earth for a long time. It's abundant now as well. And these samples, these rocks, these minerals, these other things that we study store information of all the events that happened through deep time. And the information stored in these rocks is a way for us to understand the persistence of life better so that we can see how our planet itself evolved and how we evolved with it.
Very good. Well, thank you for doing an emergency podcast on Strange New Worlds.
Thank you for having me again.
I think you're really eminently prepared for being on Radio Canada tomorrow.
Nice.
How do you feel?
I don't know. I'm excited, a little nervous too. So I hope things go fine. We'll find out. I'll let you know.
Okay, yeah. And all you listeners up in Canada, look out for Anirudh Prabhu's voice. A reminder that you can find a link to our paper in the show notes in case you're interested in downloading it and digging through the details yourself. The paper is open access and published in the Proceedings of the National Academies of Sciences. If you're enjoying Strange New Worlds, please leave us a rating or a review, or just tell a friend about the show. Next time on Strange New Worlds, I'll be joined by philosopher Christina Shakerst to talk about the linguistic, cultural, and philosophical themes in the Star Trek The Next Generation episode, Darmok. You won't want to miss it. Until then, stay safe, stay curious, and I'll see you out there.
Let me throw this back to you.
I should look it up in our paper. What exactly did we say about the sea squirt? Oh my goodness.